Hi everyone, this is the start of Chapter 11, which deals with the interactions between microbes and humans. In this mini lecture, I will be talking about the human host and infection, but mainly looking at the human host before infection. So with the human body, we're in a state of equilibrium with microorganisms. Healthy people will have a nice coexistence with microbes and they will not have disease. Occasionally, though, there may be a tip in the balance which would favor the microbes, and in this case, disease would result from that. So when we talk about a peaceful coexistence, we're really talking about the natural human biota, or microorganisms that live on or within us in a healthy way. So when we talk about infection or disease, we need to do some vocabulary here and differentiate between contact, infection, and disease. So infectious disease would be the disruption of a tissue or organism caused by microbes or their products, meaning toxins or endotoxins a microbe may produce. And that contact with that microorganism can progress to infection, and in the end of a continuum would end in disease. But keep in mind that not all contacts would lead to colonization. So you may come in contact with a microbe, but it may not be able to form a colony on your body. In addition, not all colonizations lead to infection. And in that same strain, not all infections would lead to disease. So always keep in mind that contamination without colonization and colonization without disease are the rule. More likely than not, any kind of contact will not cause disease. And part of that is thinking of the human as a habitat or a home for microorganisms. We have a normal, or what is called resident biota, and this is our indigenous flora or normal flora. And it includes an array of bacteria, fungi, protozoa, viruses, and arthropods. These are microorganisms that do not cause us harm. Presently, there is a human microbiome project. The idea of this would be to identify genetic sequences to determine what microbes we have inside of our bodies and also on the outside. Mainly because we can't culture these in the laboratory, we would need to do genetic sequences. And in this case, the project will help us determine what the normal biota plays, what role it plays in human health and disease. As we move forward, when we talk about the normal biota, we want to think about where we have this. This is not found in all parts of the body. Mainly where we find it is on the skin and in its adjacent mucous membranes, whether that's in the in the nose, perhaps in the mouth. In addition, in our respiratory tract, we have normal microbes that are within our respiratory tract. In our gastrointestinal tract, so in the stomach and intestines, we have areas that harbor microbes. In addition, we see a lot within the outer open of the urethra, the external genitalia, and the vagina. And then also thinking about our external ear canal, that's a place where we would find microbes in our normal biota. And then lastly, the external eye, so the lids and the conjunctiva. Places, though, where we consider it to be sterile, either sites or fluids. The majority of those are our internal tissues and organs. So when you think about different organs within the body and also tissues such as muscles and bones, we would consider these to be sterile. It would be unlikely to find a microbe there unless it's causing disease. In addition, we have fluids that are considered sterile. These would be fluids within an organ or tissue, including blood, urine, cerebrospinal fluid, and so on. It is when we get microbes in these that they can cause real problems. So how do we get this natural biota, this natural flora that we have? It's actually at, at birth that we have this initial colonization. Due to the fact that the uterus is normally sterile during embryonic and fetal development, it's actually the breaking of fetal membranes that first expose the infant to microbes. This could be a problem if we have premature rupturing of the membranes, and that's often something to think about when they talk about a woman's water breaking the baby, that they need to actually deliver that baby then within 24 hours or antibiotics would be needed. So how does this exposure occur during the birthing process? During a vaginal birth, the baby will become colonized with the mother's vaginal biota. In babies that are born by cesarean section, they're actually colonized by the adult skin biota. Within 8 to 12 hours of delivery, a vaginally delivered baby has been colonized by streptococci, staphylococci, and lactobacilli. And these are not considered dangerous. This is actually part of that natural flora. In addition, we have colonization of the large intestine. When we're looking at bottle-fed infants, they get a mixed population, and this is often from surface contact with the bottle, with the nipple of the bottle, and you can see that they get a mixed population here. In breastfed infants, we also see a colonization. In this case, it would be of bifidobacterium, which actually does really well because of a growth factor that's produced in milk. And this will actually help metabolize sugars to protect the infant from intestinal pathogens. So it will actually metabolize those sugars into acids, which will cut down on the intestinal pathogens. So it's a very healthy bacterium for an infant to have in their GI tract. And colonization also will continue as the infant comes into contact with family members, healthcare personnel, the environment, and food. So you can imagine all the different ways that an infant could be influenced by microbacteria. And here we see just a diagram of the origins of the microbiota in newborns. This is skin-to-skin contact through the birthing canal, through milk, through breastfeeding, and then when in a medical setting being examined. Once again, that's both environment and skin-to-skin. So hopefully this gives you an idea of the background on a healthy biota, human flora that we have, and we'll move more into the infection of microbes and also of disease.